The eyes of the jawed vertebrates, or gnathostomes, are highly homologous. The early gnathostomes evolved a larger eye, smooth muscle in the iris, an attached lens, and primitive eyelids, and these are shared in gnathostomes today from humans to sharks. The pattern of extrinsic ocular muscles which move the eye and the nerves which innervate them have been kept standard since the first gnathostomes, and these are among the most conserved muscles in the vertebrate body. One of the greatest differences in gnathostome eyes would be how the lens is focused. In more primitive gnathostomes, the lens's position is moved by muscle contraction in order to focus light on the retina, while in the amniotes, ligaments from the ciliary body will pull on the lens to change its shape so that when it is pulled to be thinner, then this allows the eye to focus on light from more distant objects, while when these ligaments relax because the muscles from the ciliary body relax, the lens becomes fatter, allowing the lens to focus on nearby objects. The Sarcopterygian ancestors of tetrapods moved the orbit higher on the head as an adaptation for vision in terrestrial environments, and early amphibians continued this. Early tetrapods evolved movable eyelids and glands which could keep the eye moist in terrestrial environments. In early mammals, which were commonly nocturnal, the number of rods in the eye increased. There were also a number of minor changes. The eyebrows and eyelashes made possible by the evolution of hair, the loss of tetrapod glands associated with the eye and the nictitating membrane. Also, the iris is only composed of smooth muscle, while in some reptiles and birds, some striated muscle is there as well. As primates began to depend more on their eyes and diurnal vision, they evolved a post-orbital bar to protect the eye. The eye became larger, and the two eyes faced forward and contributed to the same visual field as opposed to giving separate lateral images of the world. While primates and higher primates would continue to use areas of the midbrain and thalamus involved in vision in more primitive vertebrates, such as the superior colliculi and lateral geniculate nuclei. The cerebrum became more involved in vision in the occipital lobe, where visual areas became larger. In ancestral catarrhine primates, these would be the old world monkeys, apes, and humans, genetic duplication of an opsin gene on the X chromosome then allowed for two opsin genes to specialize in red light versus green light, and thus the trichromatic color vision which catarrhine primates have that most mammals lack. And so, thus, when one considers the anatomy of the eye and the physiology of vision, human vision is amazingly complex, but it has evolved through a series of adaptations throughout all of vertebrate history, and then relying on the opsins and the photoreceptors which evolved in the ancestors of vertebrates.